So today I'm going to be presenting our work. This is studies that have been going on for the past 6 years about broadly neutralizing antibody development in the Protocol C cohort and what we have learned from them. So one of the favorite broadly neutralizing vaccine strategy right now is this rational vaccine design, which relies on the characterization of complex interactions between the broadly neutralizing antibodies and their target, the HIV-1 glycoprotein. Envelope glycoprotein in order to define molecular features critical for neutralization breadth, which could then be exploited for immunogen design. So while the isolation of extremely broad and potent antibodies from chronically infected individuals has revolutionized the field in the past 8 years or so, it quickly became evident also that just looking at the end product was not going to be enough, and that we would also need to understand in greater details the maturation pathway of these broadly neutralizing antibodies over time. So not only to provide insight for immunogen design and how we can elicit proper B-cell precursor or select proper broadly neutralizing antibody intermediates to push them towards breadth, but also to assess the antibody responses generated in immunization study and determine whether we are on the right track or not, and what should be our next step. So the Protocol C cohort is a large longitudinal primary infection cohort in Sub-Saharan Africa. It enrolled 613 participants between 2006 and 2011. The average follow-up is 5.5 years. And has treatment guidelines and access to care improves all over the world. The Protocol C, together with other cohorts such as the Caprisa cohort, probably represent the last opportunities to study the development of broadly neutralizing antibodies in natural infection. So we first screened the Protocol C participant for development of neutralization breadth in the serum, and we found similar to other cohorts, that the breadth developed between 2 and 4 years of infection in about 10 to 15% of infected individuals. So we first conducted a statistical analysis of factors associated with the development of neutralization breadth and found, like many others before us, that viral load was the strongest correlate of breadth. In collaboration with Shane Crotty's lab, we also showed that the development of breadth was associated with higher TFH frequency, higher CXCR13 titers in the periphery, as well as higher anti-envelope IgG binding responses in general, all indicative of higher GC germinal center activity. So finally, we also observed that subtype C infected individuals and HLA-A3 negative individuals were more likely to develop BNAbs than subtype A infected or HLA-A3 positive individuals, respectively. All these factors overall support a model where increasing the odds of interactions between rare and possibly low affinity broadly neutralizing antibodies, precursor and intermediates with the relevant envelope variants by increasing antigen loads, envelope diversity, maybe increasing the antibody repertoire, which would also limit the landscape for viral escape itself, would promote immunofocusing. So we then mapped the broadly neutralizing antibody specificity on the top neutralizers in Protocol C and found that the majority of broadly neutralizing antibody responses targeted the V3 high mannose patch, also called the N332 glycan supersite, followed by the V2 apex region, while broadly neutralizing antibody responses to the CD4 binding site or the EMPER were quite rare. Most of those individuals did make antibodies targeting the CD4 binding site and EMPER, but they were just not broad. Indeed, a structural constraint to access these two epitopes in particular are much higher than the V3 high mannose patch or the V2 apex, which have been shown to bind antibodies which have been shown to bound the envelope trimer with different angle of approach. So we selected several individuals with different specificities for coevolution studies, and today I'm going to present results for 4 of these donors listed here, and roughly in this order. So antibodies were isolated either by FACS sorting or high-throughput functional screening of single B cells, microculture, because this work was started before the development of those now wonderful soluble native trimers that we can use nowadays to isolate quaternary-dependent antibodies. So first, with the PC64 donor, we isolated antibodies at 4 different time points and identified a lineage sharing V2 apex antibodies characteristic, namely the long CDRS3 of 25 amino acid long, which was also rich in tyrosine and was also very anionic. They also shared VH3 and DH3 gene segment that is quite common in this BNAb class. We also performed next generation sequencing of memory B cell, and we could retrace the full maturation of this lineage all the way back to the least mutated common ancestor, which was 99.6% identical to germline genes from the database. From the serum, we also obtained more than 100 envelope clones from different time points, but we also developed this next generation full-length envelope sequencing with our collaborator at UCSD, Ben Merle, which gave us an unprecedented breadth and depth in the envelope analysis. So this is quite a complex figure. We tried to put all these antibodies and envelope together and did functional analysis of their interaction. So here I'm trying to show all the data we have for this donor. In black here is the plasma heterologous neutralization as detected during our original studies. And then the dotted line here represents the frequency of the PC64 antibody sequences in the periphery, whereas the antibody diagrams represent the time where we isolated the actual monoclonal antibodies. Here at the bottom is the frequency of variants, envelope variants over time. And then we really focused on the diversity at the epitope sites, namely the residues 160, 166, 167, 169, and 181, which we showed were really critical for the maturation of this lineage over time. So mutations at position 166, 167, and 169 in particular drove the maturation of this lineage. And what is really interesting here is that this is very similar to what was observed in the CAP256 donor, which also developed V2 apex protein neutralizing antibodies. And this suggests that a localized diversity at the epitope level is actually driving neutralization breadth. What is also interesting is that these residues are quite involved in the maintenance of the trimer conformation through contact of the different protomers at the apex here. Interestingly too, the PC64 virus is a subtype A virus, while the CAP276 virus is a subtype C. And the PC64 virus, the infecting virus, started as a very sensitive virus like tier 1A, whereas the CAP256 virus started as a tier 1B. And this is something Penny and I are currently collaborating on to understand a little bit better, because this is really interesting for immunogen design, we believe. But understanding the maturation is important, but understanding the elicitation of the lineage is very critical for immunogen design as well. So we wanted to understand how the early antibody bound the early envelope, and we found these LMCA antibody that we identified in the NGS dataset didn't neutralize any of the early variants. But for when Sasha Merle from the Wilson lab crystallized the antibodies, so the LMCA is in orange, so it's the early antibody compared to the late antibody 35B, you can see that the CDRH3 conformation is much more flexible, which could explain the absence of neutralization. And for B-cell activation, you don't necessarily need neutralization, you only need binding. So we thought to test this hypothesis, and we designed this soluble trimer, and which verified in collaboration with the World Lab that they adopted a native-like conformation. And when we tested them for binding to our antibodies, as you can see here, so each line represents a different variant over time. So the ones we are really interested in here were the gray line and the blue line here. And as you can see, the early antibody didn't show any binding to any of the variants tested, even the early ones. In contrast to the late antibodies, which could neutralize, which could actually bind the variants that they correspondingly neutralized. However, when we produced these SOZIPs in 293S cells, we saw a dramatic increase of the on rate, the binding on rate by the early LMCA. The off rate was quite fast, which could probably explain the lack of neutralization, but this suggests a critical role of the glycoform heterogeneity in the activation of the germline, which is a very, very critical information for immunogen design. So currently, chemo in Andrew Ward's group and Jim Paulson at Scripps are trying to really obtain more glycan data over time in this individual. So now I'm going to change gear a little bit, talking about the two lineages targeting the N332 glycan supersite. And the PC76 coevolution study was published last year in Immunity. And we have a very similar coevolution study that is almost complete now for the PC39 donor. And what was really interesting for us is the take-home message is that we found that these two lineages shared both the VH434, but also the VK320 gene segment. They both had 22 amino acid CDRS3. However, while the PC76 lineage didn't show any indels during maturation and adaptation toward breath, PC39, the maturation of PC39 was extremely complex with different CDRS3 insertion of various length over time. So PC76 antibodies look more like 122 antibodies, whereas the PC39 antibodies are more like 135 structurally. So this is just to illustrate what I was just saying about the maturation that is really quite different between these two donors. For the PC76, you have a very dramatic expansion at very early time points. So this very vertical tree that we see here, and then after you have some kind of structural convergence to achieve breath in the very same structural way. Whereas for the PC39, you have a more moderate expansion at the beginning and then a crazy diversification afterwards with very different structural pathways of and solution to for same functional problem, and they all ended up being broad in the end. What was really also complex in this donor was the evolution of the envelope, because this donor was infected by two different variants from the same partner, which recombined pretty quickly and then segregated again into two branches that never recombined afterwards. So this makes the study of the escape and maturation pathway very complicated for us. However, if we focus on the contact residues, both the PC76 and PC39 were highly dependent on the N332 glycan as well as the N301 glycan, and they also had a very important contact with the GDRI motif here in the V3. The V1 loop was also pretty important for neutralization for both of these lineages. So in PC76, we were able to map the mutations that drove the maturation. Here for at position 328 for PC, that is on the CD4 binding site face of the V3. And then also this kind of switch, on-off switch of the glycan at position 335, which might impact the spatial position of the N332 glycan itself. For the PC39, so far what we can observe just by alignment and functional data is that the escape occurs through mutation in the GDRI or in one arm, or deletion of one residue before the GDRI, which likely recess the V3 loop and limit that contact to the antibodies. Sasha Merle is now conducting structural studies more deeply, and she has this really interesting complex structure, which we're hopefully gonna be exploiting to figure out exactly which mutation in the PC39 envelope were responsible for acquisition of breadth and driving the mutation, the maturation of the antibody lineage. So now, finally, I'm going to talk to you about our more recent data on the CD4 binding site lineage that we identified in in the PC63 donor. So this donor was one of the few individuals in protocol C that developed breadth quite late at 60 months post-infection. But this was not exactly a surprise when we found out that it was CD4 binding site targeting, because CD4 binding site antibodies have been notoriously known to be highly somatic hypermutated, suggesting a very complex and long pathway of maturation over time during infection. So we sorted antibodies at the time of breath at 78 months post-infection using GP140 YU2 probes, and we identify a really dominating VH1-2 lineage, which shared really important VRC01-like characteristics, such as a short CDRL3 with really good alignment with VRC01 itself. We tested these antibodies for breadth, and then this is the results on a 37 virus panel. And as you can see, the breadth is quite impressive. We have almost, we're reaching 90% of breadth at less than 50 microgram per ml. But even at 1 microgram per ml, we still have minimum 50% of neutralization for most of the antibodies in this lineage. So we picked randomly one of these good antibodies, and we compared it to VRC01 on a larger virus panel of 134 pseudotype envelopes from different clades, and found that, as you can see here, the P2C6 antibody is just as broad and potent as VRC01, although it has only 14.3% somatic hypermutation, compared to about 25% in VRC01. So this was really encouraging, and then it's kind of supported the work from Joe Jardine in Bill Schiff's lab, who showed that among all the mutations depicted here as hot pink balls on the variable domain of VRC01 here, only a third of them were really critical for acquisition of breadth in VRC01, and mostly those were contact residues, either directly with the CD4 binding site pocket itself, or adaptation to the N276 neighboring glycan. So here at the bottom, we also did this work for the 12A21 antibody. And in blue and yellow are the residues that were critical for breadth in both these two antibodies. And when we align our PC63 antibodies to these sequences, you can see that all the mutations that were acquired by the PC63 lineage are virtually all of them are the mean mutations that are necessary for breadth. In this paper, Joe showed that the mean antibodies for VRC01, it increases the autoreactivity potential of these antibodies, whereas for 12A21, it reduces actually the polyreactivity of this antibody. So we are curious to know what was the autoreactive or polyreactivity status of our lineage here, and then we tested them against a panel of heterologous and auto and heterologous antigen. So here you can see that most of the antibodies from this lineage were quite autoreactive and polyreactive on this particular panel. So this is not necessarily good news for vaccine design, but some of these antibodies still were actually quite good and then didn't show any polyreactivity at all. So it's going to be now very interesting to look what's the difference between these ones and how can we avoid polyreactivity during maturation. Again, the CD4 binding site is surrounded by glycan, and we know from previous studies that adaptation to the N276 glycan in particular was critical for VRC01 acquisition of breadth. So we tested single glycan mutants on 7 different strains of different subtypes, and we tested neutralization of our PC63 antibody and compared that to VRC01, PGT121, and 151 as controls. So here this graph will show the full decrease in IC50, which represent an increase in neutralization. So that means that the glycan was actually a hindrance for the antibody access. And the fold increase here at the bottom represents a loss of neutralization, which means that the glycan might actually be a contact for the antibody. So these are the results. So there are lots of dots everywhere, but what you can just look, for example, for the 448 glycan, the results are really compact. They are not as spread out as the 276 glycan, for example. For the 276 glycan, it's actually quite striking that depending on the strain, the effect can be night and day. So for GRCSF and IAVI-C22, you actually have a very reduced neutralization, whereas you have an actually increased neutralization for the other strains. And then this is quite different than to VRC01, which was consistently improved if you, neutralization which was improved if you remove this glycan. So there are really subtle variation of glycan dependency, and this is going to be very important to characterize for immunogen design, especially moving forward for a boost immunogen. So for immunogen design, again, what is really important is understanding how these somatic kappa mutation happen. Was there a special order? Which one do we want to elicit first? And are there some pathways that are more favorable than others? So we turn to next generation sequencing, and this is the work from Brian Briney in Dr. Burton's lab at TSRI. And as you can see here, so the frequency of PC63 sequences is shown here with this plain line in green. The first time we detected the lineage in the periphery was 42 months post-infection, and we start seeing neutralization breadth at around 60 months post-infection. So there's about a year and a half for the lineage to mature, apparently. And then if you compare that to the other donors that we have studied so far in Protocol C, even though the emergence of the lineage is further, it is later, the time between elicitation and acquisition of breast is not that different. And Brian plotted that on the graph here. So like I was saying, you know, the appearance of the lineage is really, really delayed in PC73. However, the time to maturation is not that different than the other lineage, especially the ones targeting the glycan epitope. So he also showed that the maturation progresses linearly at first and then kind of plateaus due to probably the mutation of the same residues over and over again. So even though you still have maturation, the percentage of somatic kappa mutation doesn't necessarily increase because it keeps mutating the same residues. So if you look at the tree here, this is what it looks like, and we're really pleased to see that the monoclonals nicely interspaced with the sequences found in the next generation sequencing data set. And what we are quite excited about is the identification of this sequence here. We found this sequence that was 100% identical to the germline gene as found in the IMGT database. So this is quite exciting, well, because this is one of the first, if probably the first VRC01 class true UCA germline ever found. So based on previous results, so studying this tree is going to be very critical for us, and these are really, really fresh data, so we haven't really been looking so deep in the data as much as we would like to for now, but we're going to do that. But the first thing Brian did was comparing the maturation of this lineage to the maturation of the VRC01-like responses that they had in their immunization mice model. That they published last year in Cell. And as you can see here, this is the acquisition on the y-axis are the VRC01 class mutations, the number of VRC01 class mutation compared to the total somatic kappa mutation on the x-axis. And if you just take the slope of the cloud here. So those each square represent a cluster of sequences that have the same variables. But as you can see here, the immunized mice, it almost looks like the maturation was actually faster than in this individual, which was so far the fastest that we know of for VRC01 class antibodies. So this is really exciting. That means that whatever they're doing with EOD-GT8 and their immunization regimen is somehow working. We are going on the right path towards VRC01, like broadly neutralizing antibody elicitation. So now they haven't achieved breadth yet in these mice, so it's going to be really important to compare the sequences with the PC63 lineage to see what we can change or what is going to be the next boost immunogen, what is the best immunogen to drive these antibodies towards breadth. So overall, if you try to summarize all this work, what we have learned for elicitation of BNAb through vaccination, so you have different challenges at different stages of maturation. You first have challenges at initiation and challenges at the maturation stage. And then those challenges are different for the different epitope. But here, for the V2 apex, for example, one of the major hurdles was the probably very low frequency, the naive repertoire due to the requirement of long CDRH3 sequences. However, we found that this PCT64 lineage was only 25 amino acid long, which apparently, according to preliminary data from Jordan in Willis in the Schiff lab, might be less rare. I mean, I'm not saying it's super frequent, but it might be less rare in the repertoire than the germline for other antibodies identified before. Again, understanding the glycan heterogeneity with, in this lineage and the dependency of the different type of glycan is going to be very important for vaccine design and should really be very valuable. And then we found this really interesting common evolution pattern for envelope with the CAP256 donor, which could really be a guide for immunogen design of groups. For the V2 apex antibody, before there was not really any gene restriction described, and then with these two new lineages, it's actually now 3 different lineages of N332 glycan superside BNAbs that share this VH434 germline, so there may be something we could exploit here. And then also, we showed that in PC76, for example, the acquisition of indels was not necessarily required, although even if it happens, it's not necessarily selected against during the germinal center maturation. And then there are very many different pathways to breadth, which is kind of a great news because maybe we don't have to be that specific. And then the immune system will find a way to find a solution. And then for CD4 binding site antibodies, the great work by several groups targeting the VH1-2 germline was still challenged by the idea that VRC01-like antibodies required high somatic appermutation and complex maturation, which supposedly would require very complex boosting regimens. But here we showed that the prediction from Joe was true, that there is a shorter pathway to breadth, and then this might be achievable. So overall, not only the unusual features are not always required, but also specific and localized diversity at key epitope might give us really a roadmap for the elicitation of breadth during immunization. And so common pathways of envelope evolution to define serial immunogen for N332 or V2 apex antibodies, but also the importance of glycoform and how we're going to control that when we produce our immunogen. And not only we have new insights for immunogen design, but these studies provide new tools to evaluate immunization studies results and also create knock-in mice models where we can test the immunogen and see if we can actually activate natural germline with these molecules. So now we have looked at several top neutralizers, but except for a couple of them, we only have one example, and an N of 1 is never really going to make a rule. So we need to study more individuals with shared specificity to really find common pathways of maturation and envelope evolution. But also we should look at what happened in all the other ones that didn't make BNAbs. And this should be very instrumental to, as I was saying earlier, evaluate the results of immunization study. What is encouraging and why did we fail? So now I just want to go to the acknowledgement and thank, first of all, the Protocol C participants, which some of them have been giving blood for 10 years, every 6 months. And it can be sometimes a challenge in some of these places to actually go to the clinic. So I also want to thank the Protocol C investigator on site, which are really doing a wonderful job at maintaining this cohort, retaining these people, providing care and counseling, which has, in Zambia in particular, really reduced the incidence of HIV in discarded couples, for example. And really a shout out to USAID for supporting this cohort and this work for the past 10 years, which is, as you saw, really going to be critical for immunogen design and really giving us hope right now. So this is the latest picture of the Protocol C investigator in February 2017, earlier this year. And I'm also very fortunate to be surrounded by many wonderful collaborators. And this work was originally also supported by an NIH U19 award to Pascal Poignard. Pascal Poignard has been my mentor for the past 6 years. And then this is so there's antibody evolution and envelope evolution, but there's also team evolution. PC76 work started with Dan and Nancy, and then Lorena is now following the story on PC39. We've had many talented technicians in the lab over the years, and then now Pascal has moved back to France, and he is starting with a new group of young and motivated people. And Lorena and Jeffrey still working with me in the lab. And then, like I was saying, our great collaborators, first with in the U19, Shane Crotty's group, Ben Merrill, has been extremely instrumental in studying the evolution of envelope, starting with Davy Smith at UCSD, and people from Daniels Burton's group, David for sharing many regions with us, Brian for his wonderful work on NGS antibody evolution, and now our more recent collaborators are Scripps, Ian Wilson, Andrew Ward for their structural analysis, but also we're starting some work with Rich Wyatt for trimer and design, as well as Bill Schiff for germline targeting and serial immunogen design. And then most recently, especially with Bernard Bagaya here on the bottom left, the PC63 was isolated together at Scripps as part of a training program, the UCID Advanced Vista program, that we're going to hopefully expand in the next few years with Dr. Kalibu at UVRI as well, but also with Eunice and Ndwadi and our supervisor, Edward Sanders at Camry. We are working closely now with Shane Crotty to dig a little bit deeper into the TFH regulation of body neutralizing antibody responses. And this is going to be part of a larger consortium that is very international, and we're really looking forward to it. I'm already working with Penny on the PC63 and looking forward to more collaborations. So last but not least, I want to thank the IAVI partners and funders. So thank you.